My name is Brody Lines. I'm the manager of HPC operations at TGen. My team of 3 and I manage all of the computational resources at TGen, as well as all of the associated storage, which includes the archive. I've been at TGen for almost 19 years. I've had a lot of various roles in that time, but almost from the beginning, I've been responsible for archive at TGen in one form or another. Started out on an IBM Tivoli system with an IBM tape library. I think we were on LTO5 at the time. That was migrated to an Oracle SAM QFS system, which was then migrated to an OHSM, and as I'll talk about today, eventually to a Versity ScoutAM system. TGen is the Translational Genomics Research Institute, established in 2002. We are in downtown Phoenix, Arizona, and at the moment, we employ about 350 people. TGen's mission includes exploring the relationship between an individual's genomic information and their health to improve patient care, also accelerating the development and deployment of new and less invasive diagnostics, genomically informed treatment decisions, and targeted therapies. And ultimately, TGen's mission is to make and translate discoveries into diagnostics, therapies, and treatments tailored to the unique needs of individual patients. TGen has several areas of focus, including personalized molecular diagnostics, precision therapeutic development, data science and bioinformatics, as well as emerging technologies and machine learning tools. The areas of research at TGen include many types of cancer, as you can see, as well as neurological diseases and disorders, metabolic diseases, drug discovery, and the rare childhood disorders. As an example, the TGen Center for Rare Childhood Disorders uses whole genome sequencing to study children suffering from ailments that have been difficult or impossible to diagnose. The center helps to pinpoint the causes of the rare disorders, allowing doctors to evaluate the proper drug or treatment options. And the Rare Childhood Disorders Center has had some success in helping parents find answers for their kids that are suffering from rare disease. We also have a facility in Flagstaff, Arizona, that focuses its research on pathogen and microbiome research. The scientists and researchers at TGen North focus on diagnostic, analytic, forensic, ecologic, and epidemiologic research of microbes critical to healthcare and public health and safety. During the pandemic, TGen North participated in multi-level genomic analysis on COVID-19 clinical sample sets. They also analyzed SARS-CoV-2 genomic diversity and provided information on how the virus spreads and evolves. TGen North has also worked with global partners on other deadly outbreaks involving cholera and anthrax. The data at TGen primarily comes from genetic sequencers. I've included some examples here of the most commonly used sequencers currently at TGen, the NovaSeq Plus from Illumina, the PacBio Revio, and the Oxford Nanopore Promethean. I've included the time for a full run of these sequencers, as well as the approximate amount of data generated per full run, and the cost to run one of these sequencers. Here's a look at the data that we currently have in archive, just to give you an idea of our current file types. The majority of the file count that we have is made up of file types related to the sequencers themselves, or results data from analysis. The capacity is largely just compressed tar files, which mostly contain raw sequencing data. There are various factors that drive the need for archive at TGen, and it's not just the scientists wanting to hoard data, which is a part of it, but sample rarity comes into play. Sometimes there are very limited quantities of a physical sample available for sequencing, so that data, once sequenced, needs to be protected because there may not be another opportunity to sequence that sample. Also, if you have the material to resequence, the cost can be prohibitive. As you saw on a previous slide, it can be pretty expensive to run the sequencers, and if you didn't budget for it, you may not have the funds to resequence. Also, there is a requirement potentially to provide a data set for future validation or to verify your research. And as the software improves, both for the sequencers themselves and the analysis, there may be a need to restore the data and reanalyze using the improved software. Here's a look at our computational resources, our primary cluster at TGen. A little on the small side maybe from what you're used to, but we like it. This is a Dell hardware cluster consisting of several different node types. We have the R750XA that is a mixed workload node. We have GPU and non-GPU workloads on that node. We have the R640 and 650, those are non-GPU workload, and the C4140 is a GPU node. It's currently a 106 node cluster, just over 5,300 cores. We have 120 A100 GPUs, those are all in that R750XA node nodes, and we have 56 of the V100 GPUs with a combined 81 terabytes of system memory. Our networking on this cluster is also Dell hardware, 400 gigabit core with 100 gigabit Ethernet top of rack, and then we drop a few 25 gig connections to each node. I don't have it on here, but our primary storage for this cluster, our Scratch, is a VAST storage array. I think it's a 600 terabyte array from VAST, and our other primary storage for the scientific data is all Isilon. Here's a look at our old archive that this process was about replacing. It's an SL3000 with an expansion module. We had a mix of T10KC as well as LTO8. The tape drives are all interconnected with fiber channel, and we have a pair of OKMs for encryption. Our cache is a 90 terabyte ZFS, and the stack was running Solaris. Some of the challenges we faced with this were the aging hardware. I don't remember the exact date when we put this into production. I have on here 2013, but I'm not sure if that's accurate. The large footprint is a problem for us. I don't know if you're familiar with the SL3000 with an expansion, but it's pretty big. And this is in a very small data center that's in our headquarters in downtown Phoenix. And that space is at a premium. So footprint was a problem. For TGen, it was non-standard equipment. We're primarily a Dell shop, dealing with increasing support costs as well as end-of-life products in this archive. We saw a few replacement options as we looked at our at what we could replace it with. We could upgrade the current library by adding new drives and maybe new hardware and software, but as I mentioned, space was at a premium where that library is, and that really didn't turn out to be an option. We looked into hybrid cloud with large inexpensive disk on-prem for ingest and restore of recent data, as well as Glacier for second copy, or a new on-prem solution entirely. Initially, we decided on the hybrid cloud, and we began our discovery and our POC process. As I mentioned, we would need some infrastructure on premises to help us control the costs, as well as to speed up the ingest of data and restores of recent data. We also discovered that while we could continue to use our current archiving process, restoring the cold data out of the cloud was more complicated and took more time than we were anticipating. It was a more user-intensive process than we were expecting. Also, it became difficult to predict or control costs related to the restores because of their highly variable nature. Initially, we were planning on passing the restore costs on to the data owners, which would be the individual labs at TGen. But it was difficult for them to estimate their restores, so it was difficult for them to estimate their costs related to those restores. And ultimately, it felt like we were paying somebody else to just put our data onto tape. Once we determined that the hybrid cloud was not going to work for us, Lynn Gerner, our VP of Technology Operations at TGen, reached out to her contacts at Versity, and they came on board to help us out. Recognizing the shared heritage between SAM QFS and the Versity solution meant that my team didn't have to spend a lot of time learning a new system. They could jump right in and be effective managers immediately. We had engaged Versity years ago when we were going through the SAM QFS to OHSM process, looking for a potential replacement. So we had done that POC and were familiar with Versity through that process. And it was nice to see that some of the same people we had worked with during that POC were still at Versity. And Versity was willing to work with us, even though what we needed in the short term was a little out of spec for what they initially provided. And what does that mean? Well, They provided a solution that fit our immediate needs with a plan for growth. What we had was a very short time frame because we'd already spent a lot of time investigating the cloud option, and we had a very tight budget. So the first solution that they provided, we responded with some requests. And those requests were basically that we needed a single node solution. So no HA, relatively small cache, all internal to that single node. What this meant for us was that with the Spectra Stack library that we went with, it also allowed us to do a single cabinet install, which helped with our space constraints. So the Versity solution provided us with a high-performance, cost-effective archive structured for scalability and future growth. The local nature of the data meant that our restore times were very fast. We didn't have to jump through any of the hoops required in the cloud to get our data back, so we could do restores immediately for researchers at TGen. And an integrated team of vendor partners and that OEM partnership between Versity, Dell, and Spectra meant that we had hardware that we were familiar with and we could integrate it into our typical management workflows. And they provided us with a simplified experience that aligned with our current needs but was scalable for future growth. Here's a look at our current archive workflow. Like I mentioned, all of our research data sits on one of several different Isilon stacks, although I guess they call it PowerStore now. But that's where the primary data lives. We use Starfish, a data and metadata management tool, as the management tool for all of our data movement. We provide users at TGen a few options for archiving their data. Those that wish, we create a Dropbox in their main lab environment that they can move data into, and it just gets archived automatically. Some users also utilize the Starfish interface directly to tag specific data that they want archived, and it gets moved. And my team can also do manual archiving as necessary. And then Starfish just moves all of that data into our Versity cache where it gets put on tape in our Spectra stack. And here's the current archive solution that Versity provided for us. We're currently running 2.23.1, but we're looking to upgrade to version 3, I think in the coming weeks. It is a Dell PowerEdge R760 with 128 gigs of RAM, 48 cores, just under 2 terabytes of metadata, and a relatively small 35 terabyte cache. We have a Spectra stack with 5 modules and 4 LTO9 drives. Our archiver policy is pretty aggressive, mostly because of our small cache size. We have a start age of 15 minutes and a start size of 100 gigs. We put 2 copies on tape, and then we auto-release everything as soon as we have 2 copies. Some of our plans for the future may include separating our storage pools by lab to allow us to segregate tape based on labs. Also a larger cache with longer retention periods for immediate restores based on lab needs. And more immediately is when LTO10 becomes available, migrating to LTO10 to get us that capacity increase without having to expand our footprint. And that is Versity at TGen. Thank you.